Hi everyone, welcome to lecture 3 on proteins. So chapters 3, 4, and 5 are all going to focus in on something that I talked about at the end of our last lecture, which was polymers, a group of molecules called polymers. And these polymers tend to form very large molecules. When you have a whole bunch of individual units that link together, they tend to make these very large molecules. So our next several lectures are going to focus in on a suite of molecules: proteins, carbohydrates, nucleic acids, and lipids, and they all fall into a group of molecules we call macromolecules, and I'm just going to write macros. Now, if this term rings a bell, maybe you're into fitness or nutrition and you track your macros, then yes, I'm actually talking about the very same things that you track when you track your nutrition. You know, if you're into your nutrients, you track your macros like carbs and fats and proteins. Same things, okay? And the reason that those are so important nutritionally is because they're important to the function of your body. So we're actually going to focus in on the biological functions and how your macros work. And today's macromolecule that we're going to hone in on is proteins. So let's talk a little bit about it. Now, just as kind of a reminder of where we left off, at the end of chapter 2, or at some point in chapter 2, I was talking to you about carbon. Remember, talking about carbon and how, carbon, and how carbon makes an excellent molecular skeleton because it has the ability to form 4 bonds. And so it just has a lot of parking spaces in which to have different configurations of its molecules. And one of the things that I mentioned to you is that individual carbon molecule units called monomers, with mono meaning one and mer meaning unit. So individual carbon molecule monomers can be linked together to form polymers, right? So just to kind of refresh your memory about that, poly meaning many, mer meaning unit. And so proteins are a type of polymer that are formed from individual monomers. In proteins, the monomers are called amino acids. You have probably heard of these cats, right? And then, of course, amino acids bond together to form the polymer that is proteins. Now, just as a refresher, the way that monomers link together to form polymers is because at one end of one monomer, you typically have a hydrogen, and at the other end of the monomer, you have a hydroxyl group. So see, hydrogen, hydroxyl group. If this is a hydrogen, there was a hydroxyl group on this end. So what will happen in order to link these monomers together is that the hydroxyl group and the hydrogen will interact to form H2O, right? H2O. We are losing a water molecule, right? And so you can see here that once we eliminate that, then this monomer is linked to this monomer. And here's the byproduct of water. And when I was learning this, I learned this as dehydration synthesis. Sorry, dehydration. I'm just going to put dehyde for dehydration synthesis. Your book refers to this as condensation. Same thing, right? A condensation reaction, I should say. When you get water out of something, it's condensing the water, right? Now, if you wanted to break these monomers, this polymer apart, I should say, so here you have a polymer, and if you wanted to break it up into individual monomers, then you would perform a series of chemical reactions to reinsert that water and break up the polymer, right? So you would perform a hydrolysis reaction, hydro meaning water, lysis meaning to break. All right, so that's just a review from last time. Now, proteins are made from amino acids as their monomer. That's the individual unit of the protein polymer, right? Here is chemically what an amino acid looks like, okay? Now, a few features I want you to notice. Remember, we were talking about carbon being an excellent molecular skeleton. So here's our carbon. And just like I promised you, carbon has 4 parking spots around which you can build a molecule, right? So we have the central carbon in an amino acid, and then there's some features that are common to all amino acids. So you have the central carbon, you have a hydrogen, and then at one end you have a functional group called an amino group. So this NHH is called an amino group. And at the other end of the amino acid, you have what's called a carboxyl group. It's a functional group that's a carbon double bonded to the oxygen and then single bonded to an OH or a hydroxyl right here. OH and hydroxyl mean the same thing, okay? So this is like a prototype of what an amino acid looks like. Now you're probably wondering what the heck this R is here. And R is literally a chemical shorthand for the rest of the molecule, okay? The rest. Now in a minute, I'm going to show you that these R side chains tend to fall into specific groups that dictate to us the chemical functioning of this amino acid. And you'll see what I mean in a minute. All right, so this is an idealized amino acid. But what amino acids really look like when you put them in water is like this right here. They become ionized, okay? So the first thing that I want you to notice is if we move from here to here, this oxygen, remember how oxygen likes to pull electrons toward itself? And that means it's pulling the electrons toward itself away from this hydrogen, and this hydrogen proton tends to dissociate from this this carboxyl group, okay? Because oxygen is a greedy, greedy little devil and it likes to gobble up electrons, it's hogging the electron from this hydrogen. This hydrogen then becomes a hydrogen ion or a single proton. I hope you can see that as I'm writing over it, okay? So what does that mean? That means that in effect, this has dissociated into a negative ion here and a proton here. And what did we call solutions that have a high concentration of protons? Well, we called those acids. So where do amino acids get their name? Well, we have an amino group, and this carboxyl group functions like an acid, okay? It also turns out that when you are in water and you have these protons circling about because this is dissociated, nitrogen is also somewhat electronegative and it will attract some of the free hydrogens that are lurking about in this solution here. So you end up with a molecule that has a positive charge at one end and a negative charge at the other, and this makes them soluble in water, right? Soluble, okay, in H2O, because it's polar, it's charged. Remember, water itself is polar, okay? So that's where we get the term amino acids, because when they are in water, the carboxyl group loses a proton, making it an acid, and then the amino group likes to accept that proton, okay? And we end up with a negative charge on one end and a positive charge on the other. All right, now let's switch gears and let's focus a little bit on these R side chains. Okay, now you may remember from your high school biology class that there are 20 different amino acids, okay? And you can think of these 20 amino acids as making up the protein alphabet. And why do I say that? Well, you can link these 20 different amino acids in different combinations to make different proteins. So it's kind of like using different letters to spell different words. Now, if you notice, all amino acids share that central carbon with the amino group on one end and the carboxyl group on the other, okay? So if you look across the top, those are all the same. What differentiates the different amino acids like aspartate, glutamate, lysine, blah, 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 right? What makes them unique are these individual R groups, okay? So you can see that all these R groups are chemically distinct. And depending upon what's in these R groups, it causes these amino acids to behave differently, okay? So for example, if you see an amino acid, and I don't know if y'all can make this out, but in your text, and maybe you can kind of blow this up in your PowerPoint, these oxygens have a negative charge on them, okay? What that means is they've lost a proton. So these R groups have lost a proton, they must be acids, okay? So if you see a negative charge here, that means they've lost a protons, they are acids. The opposite is true for these guys here. Do you see that this NH3 has a positive? We've got a positive charge there. We've got a positive charge there. So if on an R group you see a positive charge, that means it has accepted a proton and it is a base. Okay, so some of our amino acids have R groups that cause them to act like acids even beyond up here, okay? Some of them have R groups that cause them to act like bases even beyond what's happening up here, okay? Some of the amino acid R groups, you'll notice, have oxygen in them. And what did we learn about oxygen? Well, we learned that it was electronegative, right? It likes to hog electrons. So if you happen to see, if like on a quiz, I showed you an amino acid and it had an oxygen in it, but no charge. It didn't have a plus and it didn't have, or sorry, it didn't have a plus, pluses are over here. And it didn't have a minus like over here, okay? If I showed you an amino acid that had an oxygen but didn't have a minus, then that means that this is a polar side chain or a polar amino acid because oxygen's going to try and hog some electrons and steal some electrons from the rest of the molecule. So what we'll end up with is a negative charge here and a positive charge somewhere else. It'll be kind of a partial thing, okay? Now let's look at these guys down here in the green. Do you see all of these green R groups don't have a positive on them, they don't have a negative on them, and they don't have an oxygen. As a matter of fact, what you see a lot of here is just carbon and hydrogen, okay? What that means is that the charge on these R groups is going to be neutral, okay? Am I spelling neutral correct? I think I'm not. It is definitely difficult to spell when you know folks are watching. Here we go. Neutral. Okay, which is going to mean that they are non-polar. Now, why am I telling you all this? The R group is what actually dictates how these behave chemically. So let's write that down. The R group equals the chemical behavior ooh, come on. Of the amino acid. It is very difficult to write these tablets. Okay. Okay. So these R groups determine how these amino acids behave in the protein, okay? If it's got this going on here, then this amino acid is going to function like an acid in the protein. Here it would function like a base in the protein, et cetera, et cetera. I could envision a quiz question where I give you an amino acid and I ask you, is this amino acid functioning like an acid, base, polar molecule or nonpolar molecule. And you should be able to tell me based on the characteristics that I just laid out for you. Now, because we have this variety of amino acids that make up the protein alphabet, if you will, okay, 20 of them. Actually, I have down here actually 22. There are 2 that we don't really see in biological molecules, but that exist chemically, okay? But because there are 20 different amino acids that make up our protein alphabet, and they all have these unique chemical behaviors, what we find is that proteins are a very diverse class of molecules, okay? And I like to think of proteins as being sort of the workhorse of the cell. We're going to see so many examples as we go through this semester of proteins carrying out different functions in the cell. So I'm going to say that these are the workhorse. And this is my terminology. This isn't any sort of thing that you would see in a textbook, but I call them the workhorse of the cell because they just carry out so many different functions. I'm going to direct you down here to your notes tab. And under functions of proteins, we have things like catalysis. Okay, what is that? If you catalyze a chemical reaction, you are speeding it up. You are helping that chemical reaction to occur. So that means that proteins are going to have a function in running chemical reactions in the cell, okay? So an example of that is enzymes, right? Enzymes, they run the chemical reaction of the cell, and they are proteins. All right, so that's one really important function of proteins. Another one is structure. So up here, I've shown you a protein called collagen. And collagen is a really fibrous protein. It tends to be involved in, it's involved in what we call the extracellular matrix lots of times, but you guys know, you can think of collagen, you've probably heard of like collagen lip injections. Collagen is in your cartilage. It is a very important part of the structure of your body, okay? Movement. All right, movement, I don't have pictures of it up here, but for movement, if you've ever heard of the proteins actin and myosin, those are 2 proteins that are involved in the contraction of muscle tissue. So proteins are involved in movement, signaling. I don't have, let's see here, this is, I guess I'll put it here. This is one that binds to DNA and tells DNA when to turn genes on or off. That's kind of signaling. But another thing that proteins are involved in terms of signaling, a lot of hormones are proteins, okay? Transport, I've got here for you something called a porin, okay? That allows water to be transported into the cell. And then defense, antibodies are proteins, right? And they defend your cells against different foreign body invaders. So these are just examples of different protein shapes that I just pulled from your chapter, but I do want you to know the 6 functions of proteins, okay? What they are, so we got enzymes involved in chemical reactions, structure, with things like collagen, things that are involved in like your body's support, movement, like muscle contractions, signaling, like hormones, transport, things like pores on the in your cell membrane that let things into and out of the cell, and defense would be things like antibodies. And you don't have to memorize these various proteins. I really threw these up here so you could see how different some of these proteins can look, okay? All right, so let's talk about how amino acids polymerize to become proteins, all right? So first thing you need to know is that all proteins have what we call an N-terminus and a C-terminus. Terminus means end, right? So here's spelled out for you. And I probably should have started with N-terminus on this end. We actually start thinking about proteins from the N-terminus to the C-terminus. Where do you think we got those names, N-terminus and C-terminus? N comes from the nitrogen in the amino group, and C comes from the carbon in carboxyl group, okay? So what happens is these things line up with N on the left, if you're looking at a chemical equation, and the C on the right, and they link up, and can you envision here, we've got these two hydrogens on your amino group, and this oxygen on the carboxyl group. So what's going to happen? When you're undergoing this polymerization, you're going to lose your water via condensations. I'm just going to put con here just to make this go quickly. Via condensation, and you're going to end up with these two amino acids being linked. Okay, so one of the things I want you to know is that the bond between the carboxyl group here and the amino group here in a protein, this is called a peptide bond, okay? So make sure that you understand what the name of this bond is, this peptide bond. And what's really interesting about this peptide bond is that it is very, very stable. And that's because part of the characteristic of this bond is that there is an electron sharing between carbon and nitrogen. It's kind of unique. They like to pass electrons back and forth. And because they do that, even though this is a single bond, it behaves like a double bond. So this is a very stable bond, okay? Now, one thing I want to point out to you, because you may read it if you ever come across an article for like a research article, or if you're reading your book, once you go from being individual amino acids in your cellular solution and you've linked them up to be part of a protein, now we don't call them amino acids, we call them residues, okay? So let's just say we've got an R group here. Let's just say that, let's go back here. Let's say that that R group corresponds to a lysine, okay? Lysine's right here. So I'm gonna go back here. Let's say that this R group corresponds to, makes this a lysine amino acid, okay? So it's got all the characteristics of a lysine. Here we're gonna call it an amino acid, but once we've got it linked up and it's involved in becoming a protein, we don't call this lysine an amino acid anymore, we call it a lysine residue. It's still the same thing, it's just that now it's part of a larger molecule. It's not floating around on its own, okay? Now, a couple things I want you to notice. Proteins, first of all, we're gonna go through, proteins have what we call primary, and the abbreviation for primary is 1 with a degree, secondary, tertiary, and quaternary structures, okay? So we're going to go through each of these structural types on a protein to understand, on proteins to understand how they are built as molecules. So what I just showed you here is the first step in making a protein, where we link up amino acids, okay? So now we've linked up these amino acids, but actually we don't have a protein yet. What we have is the primary structure on our way to becoming a protein. What we have is actually a polypeptide chain. Polypeptide chain, okay? Because we have multiple peptide bonds. Can you see that? So here's a peptide bond, there, boop, boop, boop. A couple of things to notice about this polypeptide chain. First of all, it's a chain of amino acid residues now because they're linked together. They're not individual amino acids, all right? Another thing to notice, can you tell that we have a backbone going along one side and all of the R groups are along the other side? And so this allows the R groups to interact, okay? Remember I told you those R groups all have unique chemical characteristics, and we want them to be able to interact chemically with one another. And so when a polypeptide chain is formed, you have a backbone on one side, the R groups on the other, and that's going to allow them to interact with one another. Another thing I wanted you to notice is that when we talk about polypeptide chains and when we talk about proteins, I mentioned that we talk about an N-terminus and a C-terminus, And we talk about them as starting from the N terminus and going to the C terminus because that's the direction in which they are made. Is there 8? 1, 2, 3, 4, 5, 6, 7, 8. Yeah. So if you were to number them, you would number the first amino acid residue after the N terminus would be number 1, going all the way up to number 8. Okay. Another thing to notice, so the primary structure of a protein is simply the sequence of amino acids, okay? The sequence of amino acids that make up the polypeptide chain. That is the definition of the primary structure of a protein, okay? Just the amino acid sequence. And so if we look at this primary structure, we've got our alphabet that's making up our word, and you could go look at your little chart to see what these are. I am not requiring you to memorize the individual amino acids, okay? I am not interested in that. I myself can't do that, and I am a geneticist, but I can tell you that this is a non-polar side chain. I can tell you that this is a polar side chain. This is an acidic side chain, okay? Those are the kind of things I want you to know. All right, let's look at the primary structure here of this polypeptide chain. We're actually going to start talking about how proteins can fold up to develop what's called a secondary structure. And the reason that these primary polypeptide chains can fold up is because they have flexible bonds. Now, it turns out that the peptide bond here, you see how it's pointing to it here, and it's, I think, in blue. All these are blue peptide bonds. So that's a peptide bond. I think that's a peptide bond. Okay. That is actually not a very flexible bond, but the bond on either side of the peptide bond is flexible, okay? And so what that's going to do is it's going to allow this polypeptide chain, this primary structure, to fold. And so what does that folding look like? Well, that folding of the primary structure results in what we call secondary structure. So if you think of the primary structure as just being like a string of beads, where I come from, we celebrate Mardi Gras, so I think of these as Mardi Gras beads, right? If you have flexibility here, you can fold these beads up like that, right? So it's just beginning to fold up a little bit. It's no longer simply a straight set of beads on a string, it's folded up. And so there's two different ways that these polypeptides can fold up. They can fold up in a spiral configuration, almost like DNA, okay? That's called an alpha helix. Or they can fold up kind of like what I showed you here, almost like when you were a kid and you would fold up paper to make a fan. This is called a beta sheet, or sometimes called a pleated sheet. And the reason that this happens is because those bonds were flexible, okay? So primary structure, just to reiterate, is just the sequence of amino acids, all right? Secondary structure is when we start to let those bonds be flexible and fold up in on themselves. So then let's talk about what tertiary structure is. Tertiary structure is when we start letting the, there we go, is when we start letting the side groups interact, okay? And I'm going to skip to the next slide because I think it actually will back up. I should probably have reordered these. Let's think about tertiary structure this way. Okay, this is your secondary structure right here. Remember me telling you that there was a secondary structure that was a helix that looked almost like DNA? I like to think of this as a telephone cord. I know that we're also used to mobile phones, but maybe in your grandma's house, you've seen one of these old-timey telephone cords that looks like this. A lot of times, if you've seen this, I use hair ties that look like this now, okay? But you've seen these, right? So this would be like the secondary structure. If you had it flat, that would be primary structure. got it all twisted up into a helix, that's secondary structure. Now, if you use this helix to make a knot, that would be tertiary structure. So tertiary structure is when this secondary structure starts to fold in on itself to make sort of a larger knot looking thing. Now, what causes this knot to form are the interactions of the R groups. That's what causes the knot. I'm going to put causes. So let's take a look here. If you look at this hypothetical protein, what we have here, the flat surfaces represent sheets, the beta sheets, even though they're not pleated. Just know that that's how your book is representing the sheets, okay? The beta sheets. The beta sheets. And then these, of course, are the alpha helix shapes. Okay. So let's just say we've got our pleated sheet and our alpha helix, and let's just say that we have a polar R group here and a polar R group here, and they can interact with one another via hydrogen bonds, right? Here's another one that's interacting via hydrogen bonds. So you can see that there might be a magnetic attraction, and that might cause a knot to form here, okay? There are other interactions called, okay, this one says hydrophobic interactions. This is nonpolar, and this is nonpolar. Now let's think about that. If you've ever had the experience of making salad dressing with like oil and vinegar and water, and you know that the oil kind of clusters together, right? Because it doesn't dissolve in the water. It forms little globules. Another way of thinking of this is if you were to take like melted candle wax and drop it in water, it forms a globule, right? And it's because the oil and the water are not soluble, right? The oil doesn't dissolve in the water and it tends to stick to itself. So here we have an interaction where you can think of this as almost like two oil molecules because they're nonpolar and they're globbing together to interact with one another because they don't dissolve in the water. Here's an example of an ionic bond. Now, remember me telling you that ionic, we have a negative and a positive, right? So these are two ions. And again, we have an electromagnetic force where negatives are drawn to positives, okay? Here is something that we haven't talked about. This is actually a covalent bond called a disulfide bond. And it's just another interaction between the R side groups on your amino acids. This is actually what causes you to have curly or straight hair. If you have a lot of disulfide bonds in the keratin of your hair, guess what? You have curly hair, okay? So all of these things cause the protein to form a knot, and that knot structure is the tertiary structure. So we're just gonna say the knot structure, okay? That's just an easy way to remember it, but it's the protein secondary structure folding in on itself as a result of the interaction between R groups or side groups, okay? Okay, so what's really important about this tertiary structure is that this is the function. Let me see, where can I write this so you can really see it? I'm gonna write it real big right here. This is the functional structure. Now our protein has the shape that it needs to do its job. This is the final product. Now we have a true protein. Whoops, okay. So once you have this tertiary structure, you have the functional structure of the protein, and you can actually call it a protein now. Up until now, it's just been a polypeptide chain, okay? So this is the big deal. You want to get to this tertiary structure for the protein to work, all right? I'm going to just scooch ahead to show you what I mean. Boop, boop. Okay, so let's look here. So here's a tertiary structure composed mostly of alpha helices. Here's one that's got alpha helices and beta sheets. Here's one that's got a bunch of disulfide bonds. Let me show you this. I'm going to go down to the very end. So this is an enzyme, right? Enzymes help to make chemical reactions in your cell move forward at a rapid rate. Enzymes have to be a very specific shape so that the products, that we call them the substrates, I'm going to go over this in a second, but so that the chemicals that they're helping to interact and react with one another fit inside this little enzyme shell, okay? But that only works if the enzyme is the right shape. It only works if the enzyme has its proper tertiary structure. All right, so going back. Oops, there we go. So these are examples of tertiary structures. These are examples of functional proteins. All right. And I have in your notes, once you get to tertiary structure, now we are a protein. All right. Now, what's interesting and what should make complete sense to you, because you've studied the central dogma of biology now, and you know that DNA gets transcribed into RNA, which is a messenger molecule. That messenger molecule then gets translated into protein. And I mentioned this before, right? So now let's look at what's going on here. We have a DNA sequence, right? And in your original DNA sequence, this is the sequence of A's and C's and T's and G's. And it actually ends up coding at the end of all of this for an amino acid called glutamic acid. Now, if we're talking about hemoglobin, this is the normal configuration, right? And you end up with a protein that folds normally, and you end up with a normal tertiary structure, and you end up with normal red blood cells that carry oxygen very well, and they can flow through your capillaries very well. But if you have a mutation in the DNA sequence, like I've represented for you here, and this is in beta hemoglobin, if you have a mutation right here, as some people do, then what will happen is this DNA then ends up coding for a different amino acid called valine. This valine has an R group that causes this thing to fold differently. That different folding results in a differently shaped protein that ultimately causes you to have blood cells that are shaped like a sickle, and they don't carry oxygen as well, and they don't flow through your capillaries as well, okay? So this is a direct example of how DNA gets changed into protein, and how when you change the DNA, you change the characteristics of the protein, and you can end up with a very different result, can't you? All right. Now, we talked about tertiary structure, and for most proteins, tertiary structure is where they stop. They're functional after they have all that folding and knotting up. But there are a few proteins where you have individual units that have a tertiary structure, and they bind together to make kind of like a mega protein, right? So each of these is actually an individual protein. It's coded for by an individual stretch of DNA, okay? So this is a protein that has its own tertiary structure. This is a protein that has its own tertiary structure, et cetera, et cetera. But for this particular protein called hemoglobin, you've heard of it, right? Hemoglobin is the thing in your blood that transports oxygen. In order for hemoglobin to work properly, it needs all four of these individual proteins to work together to become the functional unit. And so quaternary structure is when you have to have multiple individual proteins bound together. Okay, So I have down here, quaternary structure is a combination of multiple protein subunits into a single functional unit. All right. This one's another example. It's a coprotein. This is a dimer. Di means two, mer means units, right? Hemoglobin is a tetramer. Mer means units, tetra means four. 1, 2, 3, 4. Okay. All right. What else do I have for you? So I like this. It's a nice overview of the levels of protein structure. Know this, okay? I always ask about it on exams, may ask about it on a quiz, okay? Make sure that you understand what these various levels of protein structure are. Make sure that you can tell me why tertiary structure is important, okay? Why is it important? What is quaternary structure and what's an example of it? All right? Now, let's go back to that slide that I showed you when I was talking about tertiary structure, okay? So this was that tertiary structure slide. And I've duplicated it for you here. And remember that the thing that causes the protein to take on a tertiary structure, which is its functional shape, right? The thing that causes that to happen are all of these interactions among the R group side chains. But notice that these interactions are often things like hydrogen bonds, right? Hydrogen bonds, you can think of them as being like magnetic bonds. And just like when I talked about water, I told you that hydrogen bonds can be transitory, right? They, you know, if you have a magnet, that's not a permanent bond. You can stick them together and pull them back apart and stick them together and pull them back apart. Ionic bonds are similar to that, right? They are an electromagnetic attraction. So you could stick it together and pull it back apart. Well, if you heat this thing up, what do you think is going to happen to some of these hydrogen bonds or ionic bonds, right? What if you added an acid which has a bunch of positive charges in it? Do you think that might mess up some of these interactions? And of course, the answer is yes. If you add heat, if you add acid, if you add bases, you can disrupt the bonding. Disrupt the bonding of the tertiary structure. Okay, so heat, acid, bases will do that too. They can disrupt this bonding of the tertiary structure and they cause the protein to denature, okay? Or I have it as denaturing, but denature. Another way of thinking of this word denature is just unfolding. It causes this protein to unfold, okay? Now, the problem is, if you unfold this protein, you've taken away its functional shape. This protein is no good to you anymore. If you want to think about a real-world example of protein denaturing, think about frying an egg. Okay, when you crack open an egg, you've got the egg white and you've got the egg yolk. The egg yolk is mostly fat and cholesterol. The egg white is protein. Now, what happens when you put that egg with its slimy egg white and its yolk into a frying pan? Well, the egg white is called the egg white because it goes from looking like something that's kind of snotty to looking like a hard white substance. And the reason that happens is all of the proteins that were all folded up, when you heat them up, they become unfolded and they start to like get all tangled up with one another, and that's what causes that to become rigid, okay? So that's a real world example of proteins denaturing. So I told you that this happens because of heat or pH, and so this is an example of it. So here we have a protein called ribonuclease. Don't worry about what it is or what it does. But I just wanted you to see, like here is its functional shape. You've got hydrogen bonds here, you've got disulfide bonds, and you have all of this resulting in the unique shape that is the functional version of this protein. But let's say that we added acid here, okay? Now what do you see? You have the primary structure is still there, but you've unfolded this bad cat, haven't you? And it's not going to be functional anymore, okay? So What if that happens inside your body, right? Sometimes proteins actually, this can happen in your body. It happens when you get hot, like when you maybe have a fever or if you have a burn or something like that. Well, it turns out that we do have a way to repair proteins. So I'm going to put protein repair. So we do have a way to repair proteins, and that involves the use of something called molecular chaperones, or many of these molecular chaperones are called heat shock proteins. And this name should make sense. If your body experiences a burst of heat, it might cause your proteins to unfold. So here you can see we have some unfolded proteins, and it says it's a non-functional aggregate. So let's just imagine a situation where you have gotten hot, almost like with the egg white example. You've gone from being a slimy sort of substance to the proteins unfolding and they're starting to link up. Well, before that happens, what will happen is your body will use these molecular chaperones or these heat shock proteins. Now let's think about what a chaperone does. If you are at a middle school dance or a high school dance or on a high school field trip, chaperones block inappropriate interactions, right? So chaperones block inappropriate interactions. What these molecular chaperones and heat shock proteins do is they try to find the regions on your primary structure that are nonpolar, okay? And the reason that they do that is because remember me telling you how nonpolar like oil droplets want to like combine together to make a larger oil droplet because they're not water soluble? Well, if you unfold these proteins, notice that you're gonna have these nonpolar regions trying to coalesce together to form sort of, think of them as oil droplets because they're not water soluble. But if you introduce these molecular chaperone proteins or heat shock proteins, see them right there, and what you can see is happening is they're blocking off these nonpolar areas and leaving open the areas that are maybe polar and can form hydrogen bonds, or they can form ionic bonds or something like that. And that encourages the protein to refold to its normal functional shape, okay? So that's how we can repair proteins using something called molecular chaperones or heat shock proteins, all right? So protein shape is actually flexible. So I've told you that you have to have a unique tertiary structure for the protein to work properly. But sometimes there are proteins that are floating around in the cell that remain unfolded, or they might be folded in an alternate shape to where they're not functional, okay? Just hanging out in the cell and they're non-functional. And what happens is they become activated when other molecules bind to them. Okay, let me give you an example. This is something called a sodium-potassium pump. This here, this little bit here, is a cell membrane. This is the outside of the cell. This is the inside of the cell. And for reasons that we'll talk about later, sometimes you want to build up the amount of sodium outside the cell and bring potassium inside the cell, okay? This has a lot to do with the functioning of your brain and some other cell signaling, okay? So what happens is this is a protein, okay? The sodium-potassium pump is a protein sitting in your cell membrane. Now, when sodium binds to this, notice that we have a bunch of positive charges, right? And we've also going to use some ATP. And when that happens, the positive charges on that sodium and the energy in that ATP is going to cause the folding of this protein to change. And you see we've gone from this configuration where this protein is open to the inside of the cell, positive charges, ATP, kapow. Now the folding has changed to where the opening of the protein is outside the cell. When that happens, it creates a situation where the sodiums no longer bind so well to this protein, but what does bind well are these potassiums. They come in, they encourage the protein folding to change one more time, and you can see what's happened. We've kind of gone inside out and restored the original situation, okay? So when you have a situation where proteins are activated by other molecules and it changes their shape, we call that a conformational change. We are just changing the conformation of the protein to alter its function or take it from an inactive state to a functional state. And we're going to talk a lot more about conformational change later, okay? One thing I thought you guys would find interesting that's a phenomenon that's associated with proteins and protein folding is that sometimes protein folding can be infectious, okay? There's a disease that you probably heard from by its common name, by using its common name, but it's called bovine spongiform encephalopathy. You might know it as mad cow disease, all right? And this thing is so weird. It is a disease that's caused by protein misfoldings. So here's what happens. There are some little protons called little proteins called prions, okay? And these prions, under a normal situation, this is what they look like, right? They're normally alpha helices. But every now and then you can end up with a diseased prion that takes on this shape here. And when it encounters another protein that's just like it, it creates a conformational change where this protein now takes on this altered folding, and it can go on and contact another protein and change its folding, et cetera, et cetera. So here's what happens. In the brains of some cows, they found some of these infected proteins, right? And these proteins, as they encounter other proteins in the brain, they cause these other proteins to become malformed, and it resulted in holes in the brain tissue of these cows, right? So spongiform encephalopathy. So it creates a situation where the brain tissue of these cows looks like a sponge. Encephalo means brain or head, okay? Pathy means disease. So it is a sponge-like head disease. So the proteins in the brain of these cows, they were contacting each other with these malformed prions, creating holes in the brain tissue. So what started happening, this really happened a lot in Europe and in particular in England in the late 1980s and the early 90s. What they would do to like rear cattle is they would feed cows the remains of other cows that they weren't using. So like you cut up the cows for steak and you've got this leftover parts, right? And you're like, what am I going to do with this cow brain? You know what? I'll feed it to my other cows. Well, what they were doing is they were feeding the cows infected brain protein, right? And that caused the new cows to have these diseases. Well, guess what? What happened is people started eating this infected cow tissue, okay? Because during the butchering process, some of these infected cow brain proteins would end up getting introduced into people's steak and hamburger and things like that. And people started getting this mad cow disease. And it was a result of these prions, okay? So what's really, really fascinating about this is that you could be infected with some of these mad cow prions and not know it. Sometimes you could be walking around with a mad cow disease prion that remained inactive, and suddenly, after 50 years of having it, you start exhibiting symptoms of mad cow disease. Another thing that's interesting is they can't really disinfect the way you disinfect for bacteria. Like we can't seem to kill these proteins. They have to actually just bury the autopsy materials that they use, like the instruments when they do an autopsy on someone that's died of mad cow disease. They can't heat it and get rid of the prions. They can't use disinfectant to get rid of the prions. They can't even use UV radiation to get rid of the prions. No matter what they did, they couldn't get rid of the prions, and they just had to start burying anything that came into contact with mad cow disease. So there was an outbreak of mad cow disease, especially in the UK in the late 1980s. And so what's interesting is that anyone that was living in the UK during that time, like let's just say that you're a military brat, meaning that your mom or dad was in the military and you were living in England in the 1980s, to this very day, you are prohibited from giving blood because they worry that you might have some of these dormant prions circulating in your bloodstream. And guess who falls into this category? Your very own professor of biology, Dr. Beth Jones, used to live in England in the 1980s. And so to this day, I cannot give blood because there's a concern that folks that live there may still have dormant mad cow prions. Now, that does not mean that you can complain about me to the administration as the mad cow professor, okay? Don't ever want to hear that coming from you. All right. The last thing that we will talk about before I end this lecture is just kind of circling back around to the functions of proteins. If you recall, I'm going to go back up here to the functions list, and I think I have it here. Yes. So there are 6 functions of proteins, and one that we're going to spend a great deal of time on is the fact that proteins catalyze chemical reactions. They are often enzymes. So I just wanted to mention enzymes one more time before the end of the lecture. So enzymes function as catalysts, and catalysts are substances that increase the rate of chemical reaction without themselves undergoing any permanent chemical change. And so I just want to introduce a set of vocabulary terms that we are going to come back to later, okay? The enzyme is the protein itself, okay? So that is the protein. And the reason that it can function as an enzyme is it has a unique shape that is its tertiary structure, right? So I'm just going to put that here. Now, when it is properly folded in its tertiary structure, it has a shape that has openings called active site or sites, okay? And these active sites are the perfect shape for the reactants of the chemical reaction. In an enzymatic reaction, the reactants are called substrates. So can you see that these substrates will fit beautifully into the active site of the enzyme if the enzyme is properly folded? Now, what happens is because these substrates fit beautifully into this active site here, the enzyme brings them into close contact with one another to encourage their interaction and chemical bonding, and that results in a chemical product being formed, and the enzyme is then free to catalyze further reactions, okay? So things that I want you to know is what an active site is, what a catalyst is, what is a substrate, okay? So just make sure that you're kind of familiar with this so that when we go over it during our enzyme chapter, which I believe is chapter 7, maybe chapter 8, you'll be familiar with this terminology. Okay, and here's an example of an enzyme with an active site. So here's the active site, here's the substrate. This kind of gives you a more real world look at what that would look like. Okay, so that's everything for chapter 3. This brings you to the end of the videos for this week's lecture set. Just as a reminder, for my 105.2 and 105.3 courses, this does not apply to the Darlington High School dual enrollment students, but if you're in 105.3 or 105.2 or 105.3, I had a homework assignment due at the end of this week. That has been extended until next week so that everyone has an opportunity to get their textbook websites functioning properly. Okie doke. Please let me know if you have any questions, and we can talk about all of this during our in-person session on Friday. Have a good day. Bye-bye.